The overall goal of this procedure is to prepare chromatin from adult mouse skeletal muscle.
This method can help answer key questions in gene regulation in skeletal muscle fibers. The main advantage of this technique is that it allows preparation of chip-grade chromatin from mouse skeletal muscle, a physically resistant tissue with a high content of structural proteins.
We first had the idea of this method when we were seeking to characterize the molecular mechanisms underlying glucocorticoid-induced muscle atrophy, which required the identification of glucocorticoid receptor DNA binding sites in mouse skeletal muscle fibers.
Demonstrating the procedure will be Shilpy Joshi, a postdoc from our lab.
To begin this procedure, recover frozen or freshly dissected hind limb muscles. Subsequently, mince the muscles in a 2 milliliter test tube containing 1 milliliter of ice-cold hypotonic buffer to a homogeneous preparation of small pieces using fine scissors. Afterward, leave the tube shaking on a benchtop agitator at 4 degrees Celsius for 5 to 10 minutes. Next, transfer the dissected tissue to a 14 milliliter round-bottom tube containing 4 milliliters of cold hypotonic buffer. Homogenize the minced tissue using a mechanical tissue homogenizer for 15 to 30 seconds. Afterward, transfer the homogenate to a 15 milliliter tube. Add cold hypotonic buffer to result in 10 milliliters of the sample and fix the homogenate before proceeding.